Okay, last speaker of the session, right, is Paul
Yep.
Klein from Novartis, huh?
Yes. Hello, everybody. Last talk before lunch break, so we can do this. I'm going to talk to you about a little bit how we're trying to model and predict patient survival, progression-free survival times from or including single-cell data. So patients are different, and we love this kind of heterogeneity. That's great and fantastic. Unfortunately, their cancers are heterogeneous and different as well. So typically, when we only have one kind of treatment, we can't hit all of the patient's tumors, and also within tumors, we can't hit all of the cells, leaving persisting disease that can eventually grow back out and cause relapse. And combining different therapies that um, will increase the chance for us to hit more patients and more cells within patients as well, leading to better outcomes. So one way to think about the diversity in patients and cells is by thinking about sensitivity in terms of the central limit theorem, where like a lot of different events on their own are like very chaotic and random with essentially unknowable outcomes. Um, in biology, you can think about this as like resistance mutations that can either be present or not present, or abundance of a target protein, which is a function of like transcription, translation, modifications, the cell cycle phase the individual cells are in, local diffusion of drug concentration, and so on and so forth, leading all taken together, these like kind of random events, to a distribution of drug sensitivity um, that has been extensively looked at by Amy Pomeroy and Adam Palmer, whom I'm working together with in the paper I'm citing here. So they like also looked at some barcoding and tracing experiments to um, actually prove that this is not just a concept, but also a real like reflects real biology, this sensitivity distribution. And now, of course, if you treat these patients, what happens is that the resistant cells die off first, and then under treatment pressure, the more resistant cells keep just growing and leading to initial responses, but eventual relapse. And what we're doing is modeling this actual heterogeneity in as differences in growth rate. So there are these growth rate metrics that are typically used to measure drug response in vitro. And when we kind of take this into account, we can integrate over this distribution, this log normal distribution as it is actually of sensitivities, and then at each time point calculate the tumor size that would um, arise from this. And we've adapted this model to continuously acting drugs as compared to Amy and Adam's paper, where they were working in blood cancer for like blasts of chemotherapy that will kill fractions of cells, whereas we are interested in like continuously acting drugs, such as CDK4/6 inhibition, which is a typical like line of therapy for HR positive breast cancer. So what this model framework allows us to do is parameterize each individual therapy agent to data that was observed, for example, in like cohorts of mice as preclinical data or in actual clinical trials. So, and then what we can do is basically add up these two effects and predict what the simulation of the compounds would do in the clinic. So as you can see here, it's like quite fascinating how we can find again or like predict or model the effect of the combination of two agents in clinical trials. Asking the question, can we actually measure this heterogeneity? So is there a way for us to not just fit everything to observed data, but get some biological measurements to put into this model? And we thought about, well, single cell sequencing is a popular method, and basically exactly doing that, measuring heterogeneity with across patients when you have like samples of different patients, but also within individual tumors when, well, you're measuring the single cells, right? And then another dimension of data that we're using is drug screening, and which is typically done in cell lines. And we have kind of the idea that we could sort of project the metrics that we measure in cell lines to the single cell data, kind of getting single cell predictions for these drug sensitivity metrics. And what we need to do to do that is measure similarity between single cells and cell lines in some way. And what a good method that measures this similarity should be able to do is identify the correct pair. So if we measure single cell data from a cell line, this method should identify the cell line again. And then the other thing it should do for if you have single cell data of patients or mice, it should identify like the cell lines it predicts to be most similar to these single cells should be of the correct tissue where the cells originated from. And we compared a couple of different methods to do that. And it turns out that, in fact, just looking at the transcriptome and doing Spearman correlation was superior to other like transformer-based methods, like, for example, GeneFormer, at both identifying cell pairs and also the correct tissue of origin. So we're actually going for this Spearman correlation of transcriptome or gene expression to measure similarity. And then we use the single cell data and drug screen. So now that we predict the drug sensitivity for each single cell, we fit then log normal distributions for both patients, and within patients, the like cell to cell heterogeneity. And we can then use these distributions as input to our models and go ahead and simulate a bunch of, in this case, mouse clinical trials where we have like collections of PDX models that are treated with different drugs. And we compare, yeah, there's like a labeling mistake. It should be drug A on top and drug B at the bottom for both sides. But anyway, so we compare the observed data in the mouse clinical trials and see kind of similar distributions when we use these data fitted to the single cell agents. And then when we take this together, we can simulate some like Kaplan-Meier type progression-free survival curves, which, at least for the most part, look pretty promising. Sort of like it tells us that we're really kind of capturing this diversity and heterogeneity that exists between the different individuals as we represent them with the mice in this case. While not all of the curves fit perfectly, it's still kind of nice. And I think what's really beautiful is then in the next step, this modeling framework allows us to take predictions of two of the agents that we have before, and by going from like single dimension normal distributions to a multivariate normal distribution, we can then just put basically the effects of the two drugs together and also simulate what the result would be for drug combinations, which is, of course, very important for us specifically because new drugs that are being developed are not developed in a vacuum, but they are added to standard of care therapies that already exist. And as I said before, combination is the way to treat heterogeneous tumors. So quite excited about that. So these data were in mice, and kind of an outlook what I'm currently trying to working on is do this also for like patient single cell data. Of course, there's a lot of data sets out there that are useful to do that. And just here as an example, I looked at pancreas clinical trial data. So in a historic clinical trial, they compared gemcitabine and the combination of gemcitabine and nab-paclitaxel. And this was actually a successful trial. So we would like to figure out or like be able to reproduce this success. This is still like why I say outlook is this is still I had to fit a dose that I as like input parameter to the model to really accurately like make these two curves line up. Haven't really figured out yet if there's like a better way of like not manually fitting this dose, but I think it's still fascinating that we're kind of capturing, based on the single cell data, the distribution and spread of the different patients in these clinical trials. Yeah, with thank you for your attention. I'd like to thank everybody who was involved in this work, primarily my mentors, Joel Wagner and Adam Honor, and my group, Oncology Data Science. And I would also like to give a shout out to the Novartis postdoc program, which, yeah, is great. And yeah, with that, thank you.
Is the Novartis postdoc program accepting applications?
It is once a year. Typically in like May, June time, they open up so the current round of applications already over, but like every year we have.
And
like,
did you go through the program?
Yes, I'm a postdoc at Novartis. Yeah.
Got it. Any questions?
I'm going to ask a question while Russ takes 10 minutes to go over there. So I guess one of the things I'm intrigued about was the similarity, the use of the word similarity there, because I didn't see a lot of similarities. But the one thing I guess I was interested in is how you're measuring burden, because tumor burden in an animal is a tricky thing to measure. So precisely how has it been done?
It's volume measurements. So
Caliper?
I think what they're doing, I haven't done the experiments. I just work with the data, but I think what they're doing is using a caliper and measuring, yeah, and calculating the volume. So measuring, I think 2 axes, and then yeah.
Because that gave you obviously some animals that responded really well and some animals that responded terribly.
Yes.
Right.
Yeah.
And so that was essentially gave you the bounds for your model fitting in terms of the diversity, because you said it was similar.
I mean, we didn't do any fitting for these models per se. Because we had the single cell data, we used this to inform the parameters.
Right. I guess my question then maybe more focused is if you compared the fit with a model that fit the actual animal data versus a model that fit the cell line,
right.
How different would it be?
I think ideally, without having done it, I think ideally not very different, because the distributions were similar that we saw with our like single cell informed models. So I guess when you do the fitting, you should also get similar distributions. I'm not sure if that answers the question or if.
I mean, I would like to see it. I guess my point is temporal dynamics, fit into temporal dynamics versus fit into a cell line response.
Okay.
Would be different.
Yes.
Right.
Most likely.
Yeah. Anyway, sorry.
Yeah. Thanks for the nice talk. So my question is, you want to move this to like human clinical trials, right? You want to be able to fit those from, modeling data. How do you envision dealing with things like heterogeneity of the people who come in terms of like what their tumor stage is, or, what micrometastases that you can or can't see that they already have, and, how, when these people progress out, there are different ways you can progress. Either the initial tumor is growing too much, or you have like, oh, I have a met somewhere now. So how do you deal with those types of things in your model?
So to answer the first part of the question, that was how do we kind of make sure like our model represents the patient population? Is that right? I guess ideally the single cell data that goes into it would kind of represent or be representative of the population of patients that are enrolled in the clinical trial, which is usually very controlled. Like they don't just enroll anyone, but they have like specific criteria. And then the second part of the question was, so what we're only measuring is sort of the one lesion growing too much aspect of the progression, and not a new lesion appeared that wasn't there at the previous scan. So yeah, we're not really, or at least I haven't come up with a way to deal with this question, but we're only kind of modeling this like growth of one lesion, and when that kind of doubled in volume, then we call it a progression event.
Okay, thank you.
Thank you.
Thanks again, Paul. Okay, thanks everyone for managing it through to lunch. Lunch